So we need to remember that enzymes are proteins, and another name for proteins is polypeptides. Since enzymes are proteins, that means there are these little pieces that make them up called amino acids. And so the amino acids are the monomers that build enzymes. Enzymes are catalysts that speed up reactions in living things, so like you. And they do this by reducing activation energy. And so if you were looking at a graph like this, you should be able to determine whether it is reaction A or reaction B that was catalyzed using an enzyme. And hopefully you were able to look at this and you can figure out that it is reaction B that has an enzyme in it. And I know that because the line is much lower. It took much less activation energy for reaction B to go from the reactants to the products at the end. So when we are looking at chemical reactions, we are starting off with the substrate and we are turning that into the product. So if you think back to the liver lab that we did at the beginning of the year, the substrate was the hydrogen peroxide, which was H2O2, and the product was water and oxygen. And the reason why this happened was because the enzyme that in this liver lab was called catalase was able to speed up the reaction and make this happen. And the only reason that this could happen is because the hydrogen peroxide is able to fit perfectly into that catalase enzyme. And for this reason, enzymes in their substrates are sometimes referred to as locks and keys. And so enzyme 1 here that you're seeing on the screen, you can see that the active site, or that area that's cut out of it, is oval shaped. And so the substrate fits perfectly into that active site. Enzyme 2 has a triangular shaped active site, and so that triangle shaped substrate will fit perfectly into that. For the third enzyme here, which of those 3 substrates do you think would fit into that active site? Hopefully you are thinking, hey, that answer is B, the hexagon, because that hexagon would fit perfectly into that active site. Enzymes work best in certain conditions, so they are a little picky. So they need a certain temperature to work the best. They need certain certain pH levels. So if you were looking at a graph like this one, you should be able to determine where or at what temperature is the enzyme functioning the best at, or what's the optimum temperature. So this graph on the right, which temperature would you say this enzyme works best at? So I would say it's probably at about 37 degrees Celsius. That is where the activity is the highest in the graph. So what you notice starting to happen when the enzyme gets a little too hot, so in this shaded area here on the graph, the activity levels are really going down. And so if you even remember back to our liver lab when we were boiling that chunk of liver and it just smelled oh so great, and you put it back into the hydrogen peroxide, nothing really happened. There were no bubbles forming. And so the enzyme, when it got too hot, it broke. It wasn't working. And when this happens, we say that an enzyme is denatured. And so denatured, the definition says active site changes shape on an enzyme, preventing the substrate from binding. And so that active site, that little area that has the perfect shape for the substrate to fit in, it's not perfect anymore. And so that substrate cannot fit. And if the substrate can't fit, the reaction is not going to happen at all. So here is the information that was at the beginning of the presentation, and we're going to break it down into simpler steps here. So enzymes are proteins made out of amino acids. Make sure you know that. Enzymes speed up reactions in living things, like you. Enzymes get stuff done. They do everything. Enzymes work on one type of substrate, so it's like the lock and key analogy. Enzymes function best in certain conditions. They like it to be a certain temperature or a certain pH level. And then the last thing you need to know about enzymes is that when they get denatured, they are changing shape and they are not working. And that is everything that you need to know about enzymes.